is following now, and we follow up to the continuation of this lecture. Right. Conversely, if bacteria are treated with an antibiotic that halts protein synthesis, RNA polymerase will slow down. Right. And number 8, some ribosomes become stalled and are rescued. Cellular metabolism is not perfect, and the cells must allow for errors. One problem ribosomes sometimes run across is defective mRNA that lacks a stop codon, where the synthesis of the mRNA was never completely finished, or whether it was mistakenly snipped short by a ribonuclease. Problems ensue. In the normal course of events, a ribosome that is translating a message into protein will sooner or later come across a stop codon. Even if an mRNA molecule comes to an abrupt end, ribosomes may be released only by release factor, and this in turn needs a stop codon. If the mRNA is defective and there is no stop codon, a ribosome that reaches the end could just sit there forever, and the ribosome behind it will all be stalled too. Bacterial cells contain a small RNA molecule that rescues stalled ribosomes. This is named tmRNA because it acts partly like tRNA and partly like mRNA. Like a tRNA, the tmRNA carries alanine, an amino acid. When it finds a stalled ribosome, it bends beside the defective mRNA. Protein synthesis now continues, first using the alanine carried by tmRNA, then continuing on to translate the short stretch of message that is also part of the tmRNA. Finally, the tmRNA provides a proper stop codon so that release factor can disassemble the ribosome and free it for continued protein synthesis. The tRNA domain of tmRNA lacks an anticodon loop and a D loop. A protein known as SMPB, not shown for clarity, binds to the tRNA domain and makes contacts with ribosome that would normally be made by the missing D loop. Clearly, the protein that has just been made is defective and should be degraded. As might be supposed, the tmRNA has signaled that the protein that was made is defective. The short stretch of 11 amino acids specified by the message part of tmRNA and added to the end of the defective protein acts as a signal known as the SSRA tag. SSRA stands for small stable RNA A, a name used for tmRNA before its function was elucidated. The SSRA tag is recognized by several proteases, originally referred to as tail-specific proteases, which degrade all proteins carrying this signal. These include the CLP proteases and the HFLB protease involved in the heat shock response. Will be seen in chapter 16, regulation of transcription in prokaryotes. Eukaryotic cells lack tmRNA but do not have a process called nonsense-mediated decay that degrades defective mRNA. Chapter 12, processing of RNA. Now this is a figure 13.28, stalled ribosome liberated by tmRNA. Binding of a tmRNA carrying alanine allows the translation of a damaged message to continue. First, alanine is added, then a short sequence of about 10 amino acids encoded by the tmRNA. Finally, the stop codon of the tmRNA allows proper termination of the polypeptide chain. Tail-specific protease, enzyme that destroys defective proteins by degrading them tail first, that is, from the carboxy terminal end. tmRNA, specialized RNA used to terminate protein synthesis when a ribosome is stalled by damaged mRNA. 9. Differences between eukaryotic and prokaryotic protein synthesis. The overall scheme of protein synthesis is similar in all living cells. However, there are significant differences between bacteria and eukaryotes. These are summarized in table 1304 and discussed in the following sections. Note that the eukaryotic cells contain mitochondria and chloroplasts, which have their own DNA and their own ribosomes. The ribosomes of these organelles operate simultaneously to those of bacteria and will be considered separately below. In eukaryotic protein synthesis, it is usually the cytoplasmic ribosomes that translate nuclear genes. Several aspects of eukaryotic protein genes are more complex. The ribosome of eukaryotic cells are larger and contain more rRNA and protein molecules than those of prokaryotes. In addition, eukaryotes have more initiation factors and a more complex initiation procedure. A few aspects of protein synthesis are actually less complex in eukaryotes. In prokaryotes, mRNA is polycistronic and may carry several genes that are translated to give several proteins. In eukaryotes, each mRNA is monocistronic and carries only a single gene, which is translated into a single protein. In prokaryotes, the genome and the ribosome are both in the cytoplasm, whereas in eukaryotes, the genome is in the nucleus. Consequently, coupled transcription and the translation is not possible for eukaryotes except for organelles discussed later. Both prokaryotes and eukaryotes have a special initiated tRNA that recognizes the start codon and inserts methionine as the first amino acid. In the prokaryotes, this first methionine has a formal group on its amino group, that is, it is N-formal methionine, but in eukaryotes, unmodified methionine is used. Initiation, elongation, and termination of protein synthesis in eukaryotes. Initiation of protein synthesis differs significantly between prokaryotes and eukaryotes. Eukaryotic mRNA has no ribosome binding site, RBS. Instead, recognition and binding to the ribosome rely on a component that is lacking in prokaryotes. The cap structure at the 5' end, which is added to eukaryotic mRNA before it leaves the nucleus, as seen in Chapter 12, Processing of RNA. Cap binding protein, one of the subunits of EIF4, binds to the cap of the mRNA. We're going back to just have a check on the table. Comparison of protein synthesis, prokaryotes and eukaryotes. Polycistronic mRNA, monocistronic mRNA. The coupled transcription translation, linear polyribosomes, no cap on mRNA, start codons is next AUG. You can see the details here. Likewise here as well. And the cap binding protein, one of the subunits of EIF4, binds to the cap of the mRNA. Eukaryotes also have one more initiation factor than prokaryotes, and the order of assembly of the initiation complex is different. Two different complexes are assembled for binding to mRNA. The first is the 43S pre-initiation complex. This is an assembly of the small 40S subunit of the ribosome attached to several eukaryotic initiation factors, EIFs. These include EIF1, EIF1A, EIF3, and EIF5. This binds the charged initiated tRNA, met tRNA, plus EIF2. The second complex, the cap binding complex, contains cap binding protein, EIF4E, EIF4G, EIF4A, EIF4B, and poly A binding protein PABP. During the eukaryotic initiation, cap binding complex first attached to the mRNA via its cap. And the side note reads, eukaryotic mRNA is recognized by its cap structure, not by base pairing to RNA. And continuing on the main frame. Next, the poly A tail is bound by PABP so that the mRNA forms a ring. This structure can now bind the 43S assembly in order to align the met tRNA with the correct AUG codon. The two structures work together to scan each codon from the 5' end. This scanning process uses energy from ATP. Normally, the first AUG is used as the start codon, although the second sequence surrounding the AUG is important. The consensus is R equal to A or G. If its surrounding sequence is too far from consensus, an AUG may be, let's get the continuation, skipped. Once a suitable AUG has been located, EIF5 joins the complex, which in turn allows the 60S subunit to join the cap binding protein EIF2, EIF1, EIF3, and maybe EIF5 to depart. Each EIF5 uses energy from GTP to accomplish this remodeling of the ribosome. Going back to the explanation of the diagram, figure 13.9 reads about assembly of the eukaryotic initiation complex. That's the illustration here. just take a briefing on that. Okay, take screenshots and let me read that detail for you. The cap binding complex includes poly A binding protein PABP, EIF4A, EIF4B, EIF4E, and EIF4G, which is in an unphosphorylated state when unbound to mRNA. ADP transfers phosphates the complex to make it competent for binding the mRNA P. The 43S initiation complex forms, bringing the small ribosomal subunit together with the RNA, tRNA. This complex uses GTP to attach the tRNA to the 40S subunit via EIF2. In addition, initiation factors EIF1, EIF1A, EIF3, EIF5, and the EIF2B guide and make the complex competent to bind to the 5' UTR of mRNA. The mRNA is recognized by the cap binding complex via the connections between EIF4E and PABP, which binds the 5' and 3' ends of the mRNA respectively. These two connections cause the rest of the mRNA to loop out. When this is established, then the 43S pre-initiation complex can attach and start scanning for the first AUG. After pausing at the first AUG, then the 50S subunit of the ribosome can bind and initiate translation. Now the next stage is elongation, as shown in figure 13.30. Of all the stages of translation, elongation is bacteria. In bacteria and eukaryotes is the most similar. As in bacteria, elongation factors work to decode the mRNA and bind the tRNA into the A side of the ribosome. Rather than EFTU and EFTH, eukaryotes use small e EF1A to deliver the tRNA using GTP hydrolysis for energy, and EF1B to replace the depleted GTP with the fresh GTP. The only difference is that eukaryotic elongation factors include more subunits. The remaining steps are the same. The peptidyl transferase activity of the 28S rRNA of the large subunit links the incoming amino acid to the polypeptide chain, then elongation factor EF2, direct counterpart to bacterial EFG, uses GTP to drive the conformational changes in the ribosome and ratchet the tRNAs from the P and A sites into the E and P sites. Elongation continues until a stop codon enters the A site. Eukaryotic termination differs from prokaryotic termination in two ways. First, rather than having two different release factors RF1 and RF2 to recognize different stop codons, eukaryotes have a single release factor ERF1 that recognizes all three stop codons. ERF1 binds the stop codon, but this does not affect peptide bond formation. Instead, ERF3, carrying a GDP molecule, binds to ERF1. GDP hydrolysis then rearranges the factors, and the final amino acid attaches to the polypeptide. Therefore, eukaryotes require GDP for polypeptide completion, whereas in bacteria, RF1 or RF2 is sufficient. We read box 1302, internal ribosome entry sites. Right. Although most eukaryotic mRNA is scanned by the 40S subunit to find the first AUG, exceptions do occur. And the sequence known as internal ribosome entry sites are found in a few mRNA molecules, as the name indicates. These allow ribosomes to initiate translation internally rather than at the 5' end of the mRNA. And IRES sequences were first found in certain viruses that have polycistronic mRNA, despite infecting eukaryote cells. In this case, the presence of IRES sequences in front of each coding sequence allows a single mRNA to be translated to give multiple proteins. The best known examples are members of the picornavirus family, which includes polioviruses, causative agent of polio, and rhinovirus, one of the agents of common cold. More recently, it has been found that a few special mRNA molecules encoded by eukaryotic cells themselves also possess IRES sequences. During major stress situations such as heat shock or energy deficit, synthesis of the majority of proteins is greatly decreased. Much of this regulation occurs at the initiation stage of translation, discussed later. However, a few proteins are exempted from this downregulation as they are needed under stress conditions. The mRNAs encoding these proteins often contain an IRES sequence. In these cases, the mRNA carries only a single coding sequence and the IRES is located in the 5' UTR between the 5' end of the mRNA and the start of the coding sequence. This allows translation to be initiated at the IRES even in the absence of the standard initiation scanning procedure. Finally, as in bacteria, eukaryotic ribosomes are recycled. EIF3 triggers the release of the 60S subunit, and then EIF1 releases the final tRNA. An additional factor that EIF3J then removes the mRNA. The components are then recycled. Elaborating the diagram, 13.3 beginning eukaryotic translation elongation. Once the eukaryotic 40S subunit complex finds the first AUG, then the remaining 60S subunit and associated factors combine to form the final 80S ribosome. 10. Protein synthesis is halted when resources are scarce. Proteins make up about 2/3 of the organic matter in a cell, and their synthesis consumes a major part of the cell's energy and raw materials. Clearly, when cells run on low nutrients or energy, they cannot continue to synthesize proteins at the normal rate. In bacteria, ribosomes are taken out of service during stationary phase or periods of slow growth. A small base protein, ribosome modulation factor RMF, binds to ribosomes and inactivates them. The inactivated ribosomes exist as dimers. When favorable conditions return, the inactive dimers are disassembled and the ribosomes are reactivated. In E. coli, starvation induces the stringent response, where the cell enlists transcribed genes that are essential for survival and virulence. Most of the genes are turned off at the transcriptional level. This response is triggered in part when an uncharged tRNA loads into the A side of the ribosome. This will only occur if charged tRNA is in short supply due to lack of energy or amino acids. When this happens, RNA, a protein associated with the ribosome during translation, starts making PPP, GPP, which binds to RNA polymerase to modulate what genes are transcribed. This response is also important to bacteria such as Mycobacterium tuberculosis, which causes tuberculosis. These bacteria are highly resistant to antibiotic treatment and the human immune system and can live for years inside immune cells. They are able to persist because of their stringent response. Recently, abscesses caused by Staphylococcus aureus and Pseudomonas aeruginosa both use the stringent response as part of their pathogenicity. Having been diminished using a peptide that targets GPP, the astringent response in the stringent response. Targeting the stringent response might provide useful tools against antibiotic-resistant strains of bacterial pathogens. Higher organisms also stop protein synthesis when nutrients or energy run low. However, they do so by inactivating the initiation factors rather than the ribosomes. Initiation factors EIF2 uses energy by hydrolyzing GTP to GDP after initiation is over. It is released from the ribosome with the GDP still bound. It then binds to EIF2B, which exchanges GDP for GTP or recycling the EIF2. In times of stress, a kinase phosphorylates EIF2 and prevents the removal of GDP. The GDP-bound form of EIF2 cannot initiate translation, and protein synthesis is halted. Some viruses have taken advantage of this mechanism and use phosphorylation of EIF2 as a way to shut down host protein synthesis. Enumerating the diagram, figure 13.31, bacterial ribosomes on standby during conditions. Active bacterial ribosomes can become inactive when the RMF proteins bind to them. The ribosomes form dimers with the 30 subunits attached to one another. When conditions are favorable, dissociation occurs. Ribosomes modulation factor RMF protein that inactivates surplus ribosomes during slow growth or stationary phase in bacteria. stringent response, decreasing transcription of non-essential genes when nutrients are in limited supply. Alright, going to the next page. This is a figure, take screenshots. And let me know right there. Recycling of initiation factor EIF2 is controlled when eukaryotes downregulate the level of protein synthesis. A protein kinase phosphorylates EIF2 by GDP. This prevents EIF2B from removing the GDP, and EIF by GDP stays locked in an inactive complex with EIF2B. Absence of active EIF2 decreases the rate of initiation. As usual, eukaryotes have multiple pathways of control. Another way the cell controls protein synthesis is to regulate the cap binding complex. When the cell is stressed or starved, mTOR target of the antibiotic rapamycin is prevented from phosphorylation. EIFs 4E, 4G, and 4B. Without these phosphates, the EIF complex cannot bind to mRNA and translation is halted. 11th point is a signal sequence marks a protein for export from the cell. Once a protein has been made, it must find its correct location within the cell. Although cytoplasmic proteins are made in the cell compartment where they belong, other proteins which do not reside in the cytoplasm must be transported. Proteins destined to be exported to the exterior of the cell wall must be exported through the cell membrane. Similar systems exist in bacterial and eukaryotic cells. Proteins destined for export are tagged at the N-terminus with a signal sequence. This is cut off after export by proteases attached to the outside of the membrane and is therefore not present in the mature protein. This is cut off after export by proteases attached to the outside of the membrane and this is therefore not present in the mature protein. The signal sequence consists of approximately 20 amino acids that form an alpha helix. There is a little specific sequence homology between signal sequences of different exported proteins. A positively charged basic N-terminus of 2 to 8 amino acids is followed by a long stretch of hydrophobic amino acids. The amino acid just before the cleavage site has a short side chain. Exported proteins have a signal sequence at the front. That's a side note. And a polypeptide destined for export is recognized by its signal sequence. In bacteria, the signal recognition protein, Sec-A, binds the signal sequence and guides it to the translocase complex in the cell membrane. The rest of the protein being exported is synthesized and follows the signal sequence into and through the membrane via the translocase. This is known as co-translational export. Since the protein is exported as it is made, the signal sequence is cut off by the leader peptidase or signal peptidase after translocation. Now the terms of importance here are co-translational export. Export of a protein across a membrane while it is still being synthesized by a ribosome. Signal sequence, short, largely hydrophobic sequence of amino acids at the front of a protein that label it for export. Translocase, enzyme complex that transports proteins across membranes. This is a figure 13.33, standard signal sequence for exported proteins. The signal sequence contains a positively charged domain containing lysine and/or arginine, a alpha helical hydrophobic domain rich in alanine, leucine, and valine, and a cleavage site preceded by a glycine or serine and followed by a proline. A reverse turn due to glycine is found approximately halfway through the hydrophobic domain. Please take screenshots. Figure 13.34. The ribosome making the polypeptide chain approaches the cell membrane. The polypeptide with its signal sequence binds to the signal recognition protein. The signal recognition protein recognizes the translocase and binds to it, allowing the polypeptide chain to begin its journey through the membrane. After the signal sequence exits the translocase, leader peptidase cuts the polypeptide chain, liberating the signal peptide. Final folding of the polyprotein occurs outside the cell. Right? Now, there are approximately 500 translocases per E. coli cell. Each cell exports about 1 × 10⁻⁶ proteins and from the cytoplasm prior to dividing. In a cell that doubles in 20 minutes, 100 proteins are exported per minute per translocase. Protein export is tenfold faster than protein synthesis. So the demand for a growing protein chain will allow the translocase to be ready for a new chain as fast as the ribosome can make it. Note that in gram-negative bacteria such as E. coli, most of these exported proteins are structural components of the outer membrane